To summarize, first, okay. Ashwati.
Yes, sir.
So can you just tell me what we have explained in the previous class?
We
And
learned about the cellular foundations of biochemistry.
what you have understood in a summarize in a systematic way.
Um, Sir, we learned what biochemistry is, and it's the study of life with biology and chemistry. And we also learned a few of the explanations of the cellular foundations of biochemistry.
Yeah, good. Then
We learned that cells are the basic unit of biochemistry, and then we learned about some of the organelles of the cell.
but before Ashwati, before explaining that, initially we started with the origin of
Yes, sir.
universe and life, and we said that initially we discussed about the how the universe formed,
Yes,
isn't
sir.
it?
Yes, sir.
uh,
Yes, we
so
discussed.
we said that the universe, the present day earth, it is formed because of the titanic explosion of hot energy rich subatomic particles. Then we said that initially the simple elements formed. Then the universe expanded and cooled. Then the material condensed under the influence of the gravity to form stars, and these stars further exploded and released the energy, and that energy is utilized for making the complex elements from the simple elements, simple atomic nuclei. Then these atoms and molecules further formed the masses of dust particles, and finally these particles accumulated and eventually formed the rocks, planetoids, and planets. Then that is the then coming to the origin of life. Then the life originated 4 billion years ago. Initially the simple microorganisms were formed. They have the ability to extract energy from the chemical compounds. Then all multicellular organisms evolved. Then we also said that there are many theories proposed in order to explain the origin of life on earth, and we have already said that the theory of special creation, theory of spontaneous generation, theory of biogenesis, theory of biochemical evolution, theory of panspermia, and deep sea hydrothermal vent theory. And of course in biochemistry we are, since we don't want to go into those details, I have already said in summary what is the exact each theory explains. And in biochemistry asks how the remarkable properties of living organisms arise from the different biological macromolecules. And then we discussed about the distinguishing features of the living organisms. We have already highlighted the living organisms have the high degree of chemical complexity, and they have various systems to extract and transform and use energy from the environment, and different components in the in the living organisms have different functions, and the all living organisms have various mechanisms to sense and also to respond to the changes in the surroundings, and they have the living organisms have the ability to undergo self-replication and also the self-assembly, and living organisms have the ability to evolve with the time. That is called, that is known as the evolution. So this all these we have already said in detail. Then we discussed about the cellular foundations. In cellular foundations, initially we said that all living organisms are composed of cells. That means cell is the structural and functional unit of the living organisms. And based on the number of cells present, these organisms are classified into unicellular and multicellular. We have already said that unicellular organisms have a single cell, and whereas the multicellular organisms have different cells, and we have already seen unicellular organisms like amoeba, paramecium, and euglena, and multicellular organisms like what we have already showed, all humans, plants, and other, and so on. And we have already explained the difference between the unicellular and the multicellular organisms. And one
important means photo. Phone.
எனக்கு எல்லாமே இம்பார்ட்டன்ட் தான்.
Then we discussed about the basic difference between the unicellular and the multicellular organisms. Then after that we have discussed about the certain features of the all living organisms in the living cells, and because as we already said that cell is the basic structural and functional unit of the organisms, therefore we have already said that all cells have some common features, that is the presence of the plasma membrane and the cytoplasm, as well as the nucleus in case of eukaryotes and the nucleoid in case of prokaryotes. And we said that the plasma membrane is a lipid bilayer, and it is composed of with lipids as well as the protein molecules, and this plasma membrane is highly hydrophobic in nature. It doesn't allow the polar molecules, only non-polar molecules or the hydrophobic molecules can cross the bilayer, whereas the polar molecules cannot cross the bilayer. And this plasma membrane provides shape to the cell, and it also regulate the movement of the molecules and ions in and out across the membrane. And many proteins that are present on the plasma membrane can act as receptors and transporters and so on. Then we discussed about the cytoplasm. The cytoplasm, that is the aqueous portion of the in the cell, and the cytoplasm has many subcellular organelles in case of eukaryotes like ribosomes, peroxisomes, and mitochondria, chloroplast, and so on. And in addition to this, the cytoplasm has many biological macromolecules, coenzymes, and enzymes, and other metabolites. In addition to this, it has many inorganic ions like sodium, potassium, magnesium, and calcium. Then we discussed about the nucleus. In case of eukaryotes, nucleus is present where the DNA is present in the form of chromatin, and whereas in case of eukaryotes, the nucleoid, that is the genetic material, is present in the cytoplasm. And then we discussed about the different functions of all these organelles. Then we also discussed about the cellular dimension, in the sense most cells have macroscopic and invisible to the unaided eye, but the animal and plant cells have the 5 to 100 micrometers diameter, whereas the bacteria is only 1 to 2 micrometers. And we also said that exactly the size of the cell is dictated by the lower limit is dictated by the minimum number of biomolecules required by the cell, because in case of the mycoplasma, and it has 300 nanometers, and whereas it has volume of 10 to the power minus 4 ml, and the upper limit is dictated by the rate of diffusion of solute molecules, the molecules across the membrane, and so on. Then all organisms are classified into three categories. That is three large groups, and we said that one is bacteria, second one is archaea, and third one is the eukarya. All these are originated from the same ancestor, common progenitor. And if you see closely here, that compared to the eukaryotes and the bacteria, archaea and bacteria has close similarities because they both are originated from here. So whereas compared to the bacteria and archaea and the eukaryote has some similarities. And then we discussed about based on these archaea and bacteria has further has subgroups based on their habitats. So if they are able to survive only in the presence of oxygen, they are aerobic. As I already said that in all aerobic organisms, oxygen acts as a terminal electron acceptor in the electron transport chain. That's what I said. In the mitochondria, the oxygen acts as a terminal electron acceptor, and that in order to make the high energy molecules like ATP. So whereas in anaerobes, instead of nitrate, sulfate, and carbon dioxide act as a electron acceptors. And this is the classification of these archaea and bacteria based on their habitats. So aerobic and anaerobic. Then we also these anaerobic and aerobic, these anaerobic organisms are also again further classified into two types: obligate anaerobes and facultative anaerobes. Obligate anaerobes are also known as the strict anaerobes, in the sense they cannot tolerate the presence of oxygen, they cannot survive in the presence of oxygen, whereas the facultative anaerobes, they can survive in the presence or in the absence of the oxygen. And then we discussed about the classification of organisms based on the energy and carbon source. So all organisms are broadly classified into two categories based on the energy source: phototrophs, chemotrophs. Phototrophs, in the sense they derive energy from the light, whereas the chemotrophs, they derive energy from the oxidation of chemical fuels. And these phototrophs are further classified, and of course these chemotrophs are further classified into chemolithotrophs, chemoorganotrophs. In chemolithotrophs, they oxidize inorganic fuels for getting the energy, whereas the chemoorganotrophs, they oxidize many different types of organic molecules to get the energy. And I think I forgot to mention at this point, in chemolithotrophs, these organisms uses they oxidize many inorganic fuels to derive the energy. That is the oxidation of HS to elemental sulfur, or the elemental sulfur to SO4 minus, or NO2 minus to NO3 minus, or Fe2 plus to Fe3 plus. So all these chemolithotrophs oxidize inorganic fuels to get their energy, whereas organic chemoorganotrophs uses various organic molecules to get the energy. And of course these phototrophs and chemotrophs both are again further divided into autotrophs and the photoheterotrophs in the same way, so on. So in the photoautotrophs, in case of photoautotrophs, they get the carbon source from the carbon dioxide. In the case of heterotrophs, they get the carbon from the organic compounds. So that is only the difference. And we have already said some examples for the all these the autotrophs, heterotrophs, and the chemolithotrophs, chemoorganotrophs. Then I think that's where we stopped. Then coming to the we just wanted to explain the structure of the bacteria and also the structure of the eukaryotic cell. And we know the most studied bacterium is the E. coli, and the E. coli has 2 micrometers long, and it has 1 micrometer diameter, and if you see this structure, this E. coli has the some bacteria has outer membrane, some bacteria has only inner membrane, as well as the peptidoglycan layer. You can also see here that this is the outer membrane, the outer membrane, and the peptidoglycan is called as a cell envelope. And the in case of gram, if it is in case of E. coli, it is gram negative, that is different. But in case of some gram positive bacteria and gram negative bacteria has a different structure. So this you can clearly see here that in case of gram positive bacteria, it has the plasma membrane that is inside, as well as the peptidoglycan layer at the outside. Whereas in case of gram negative bacteria, it has the plasma membrane as well as the peptidoglycan layer as well as the outer membrane. Whereas in case of gram positive bacteria, the outer membrane is not present. But here you can notice that the peptidoglycan is very thick, whereas in case of gram negative, the peptidoglycan is very thin. And of course, the peptidoglycan is chemically made up of with the N-acetyl glucosamine and N-acetyl myrmic acid with beta-1,4 linkages, and it is a very linear polymer. And the one NAG and NAM layer is cross-linked with another NAG and NAM layer by small peptides. As a result, this multi-layered NAG and NAM layer gives the rigidity to the peptidoglycan. And of course, we don't need to say that why it is gram positive and the gram negative bacteria. In the gram positive bacteria, in gram staining, it appears blue, whereas the gram negative bacteria, it appears red because of the counter stain safranin, and that many of you must have studied in the microbiology. And so this you can also see here how the gram positive and the gram negative bacteria appears. And you can see here, this is you can see here the gram positive bacteria has the very thick multi-layered peptidoglycan. You can, as I already said, that the peptidoglycan is made up of with N-acetyl glucosamine and N-acetyl myrmic acid. So it is a polymer made up of with NAG and NAM. And this is the plasma membrane, and here this is outer membrane, and this is the inner membrane, plasma membrane, and this is the very thin peptidoglycan layer is present. And coming to this is about the this one. Coming to the, as I already said that in case of archaea, as I already said that bacteria and archaea both are both are the unicellular, and the archaea has certain characteristic features, as I already said in the morning class. In the sense, the archaea can survive under extreme conditions or in the extreme environments. So because it has the ether linked phospholipids. So in the same way here, in the structure of the archaea also, it has the pseudopeptidoglycan instead of the normal peptidoglycan, and it is chemically it is slightly different than the normal peptidoglycan. So that is the pseudopeptidoglycan is present in case of archaea. So coming to the cytoplasm of the cytoplasm of the E. coli contains many enzymes, ribosomes, and many metabolites and inorganic ions. In addition to this, in addition to this, you can also, in addition to this, if you can see the structure, and of course it is not given here, and it also has the small circular DNA segment that is called the plasmid. I think plasmid, it has the plasmid, and the long linear genomic DNA is circular DNA is present in the nucleoid, and this plasmid has various functions as we have already studied. They have the they confer resistance to the toxins and also antibiotics present in the environment, and this is about uh, the this one bacterial cell. Then coming to the eukaryotic cells. So eukaryotic cells are, as we know, it is they are bigger than the bacteria. They have the 5 to, if it is animal cell, it is this cell size is 5 to 30 micrometers diameter, and if it is a plant, it is 10 to 100 micrometers diameter. And this, the eukaryotic cell has the distinguishing characteristics in the sense compared to the prokaryotes, in the sense they have the nucleus, and in addition to this it has a nucleus, and in addition to this, and a variety of membrane enclosed organelles, subcellular organelles present, such as mitochondria, endoplasmic reticulum, and the Golgi, peroxisomes, lysosomes, and so on. All these are present. But in case of plant cell, and vacuoles and chloroplasts also are present, and in some cells have the starch and the fat granules as well. So, and if you, in case of eukaryotic cell, the cytoplasm has the cytoskeletal proteins, and these cytoskeletal proteins are mainly make the three-dimensional meshwork, and these, there are three types of cytoplasmic proteins present here, and these cytoplasmic proteins are, one is the actin filaments, cytoplasmic proteins, one is the actin filaments, second one is the microtubules, third one is the intermediate filaments. All these all these cytoskeletal proteins present in the cytoplasm provides the structure and uh, mm, organization of the cytoplasm and the shape to the cell. So they are present in the cytoplasm. And now coming to the eukaryotic cell, in eukaryotic cell has the ability to build various supramolecular structures from the monomeric units, and you can see here all these supramolecular structures like a chromatin, plasma membrane, cell wall. All these are built with the monomeric units, and and of course these monomeric units, the eukaryotic cell has many monomeric and the monomeric units as well as the macromolecules. The monomeric units mainly the amino acids, nucleotide, and monosaccharides, and so on, whereas the macromolecules mainly proteins, enzymes, nucleic acids, the glycans, and so on. And so by using these the macromolecules, the eukaryotic cell has ability to form supramolecular assemblies like chromatin, plasma membrane, and so on. And of course these the supramolecular structures are stabilized by various non-covalent interactions. And of course, since the eukaryotic cell has many subcellular organelles, and these subcellular organelles can be separated by a special technique called differential centrifugation. I think we know when the cell is when the cell is disrupted, what we get is the cellular lysate, and when the cellular lysate is subjected to the centrifugation, and by differential centrifugation at a lower speed, the high density the subcellular organelles would sediment like cytoskeleton and plasma membranes and so on. And when the supernatant is removed and is further subjected and spined at the further high speed, and again the pellet contains the mitochondria, lysosomes, peroxisomes, and further when the supernatant is removed and further again centrifuged at a higher speed, again we get the microsomes such as the fragments of the endoplasmic reticulum and so on. So that means if we want, we can separate these subcellular organelles present in the eukaryotic cells by differential centrifugation, and this is about the cellular foundation of biochemistry. In summary, this is the summary of the cellular foundations. All cells are bound by a plasma membrane. We have already said that this plasma membrane has various functions, and it provides the shape to the cell. It also involved in the regulation of the cell volume, and in addition to the plasma membrane, it has the cytosol, and the cytosol has many metabolites, coenzymes, inorganic ions, and other enzymes. And also the DNA is present, the genetic material is present in case of eukaryotes in the nucleus, whereas in case of bacteria and archaea, it is in the nucleoid. And all these organisms require a source of energy to do various work, and the phototrophs get their energy from the sunlight, whereas the chemotrophs get their energy from the oxidation of various fuels, either organic or inorganic. And as we already said that if they are using the inorganic fuels, they are known as the chemolithotrophs. If they are using organic fuels, chemoorganotrophs. Then coming to the structure of the bacterial and archaeal cells also contain the cytosol and the nucleoid, and also the circular DNA segment that is known as the plasmid, and also the cell envelope. Eukaryotic cells have nucleus and various subcellular organelles are present, and these subcellular organelles can be separated by differential centrifugation. And the cytoplasm has various cytoskeletal proteins, like as I said, and all these cytoskeletal proteins like actin filaments, microtubules, and intermediate filaments, and they provide shape to the cell and rigidity to the cell. And in eukaryotic cell, the various supramolecular assemblies are formed from the other biological macromolecules. These are chromatin and the lipid lipid membranes and so on. All these supramolecular complexes are held together by the various covalent interactions. So this is this is about the cellular foundations. In tomorrow's class, we will be discussing with the other topic, that is the chemical foundations as well as the physical foundations and genetic and evolutionary foundation. We will try to complete in tomorrow's class.